Dr. Andrea Cox, professor of medicine at Johns Hopkins University. She first studied virology and has her PhD from neighbor University of Virginia, and now she's at Hopkins, where she is specialized in viral hepatitis center. She's a member of the Division of Infectious Disorders. We are in between Virginia and Baltimore. So here at the NIH, we have supported her studies, and she's going to discuss these two papers and bring her views of current science. Go ahead, Andrea. Thank you so much for helping us.
Thanks for the invitation to moderate and also for those two fantastic talks. Maybe I'll start. Jackie, you made the really important point that HCV antibodies are negative in the acute phase of infection, and HCV RNA is critical for testing for acute infection. I want to just make another point, which is Dr. Compton pointed out that reinfection occurs in individuals who've been previously treated, and we see people with positive antibodies who've spontaneously controlled HCV, as well as those who've been treated. So we've had a lot of misinformation imparted to patients based on a positive antibody test that doesn't reflect their current situation from an infection standpoint. And also HCV RNA is critical for monitoring for reinfection in individuals, including people who inject drugs who are at ongoing risk of HCV infection. So in addition to the time it takes to run the test, there's probably a cost barrier, because otherwise I think, HCV RNA testing, I would love to see us do away with HCV antibody testing and only test for HCV RNA, which is really what matters. So could you talk a little bit about maybe the cost barrier as well, or other things that you think will limit uptake of the systems that test just for HCV RNA?
Yeah, and the cost is very much a concern. I think that right now, even the enzymes that do that nucleic acid amplification, they're not particularly cheap. And in comparison to buying antibodies, which is a really well-established market that exists. So there's certainly cost issues in the assay itself before you put the device packaging around it. And then we've been looking a lot at that packaging, and the technologies that are doing that are mostly injection molding, which is just generally more expensive than roll-to-roll paper, which newspapers are printed on for really cheap. So bringing that cost down is going to be really critical to access.
Well, I hope someone considers sending patients to the doctor who are antibody positive and RNA negative, and the cost of that, and considering cost, as well as the psychological impact of telling someone who thinks they're cured of Hep C that they have hepatitis C virus infection, which I think shouldn't be underestimated.
That would be a great model to do.
I can ask a question of Andrea as well. You mentioned the maternal or parent-to-child transmission of hepatitis C is becoming increasingly a problem. I'm interested in your thoughts on risk factors for that, particularly the fact that people of childbearing age who are acutely infected in pregnancy, they tend to have higher HCV RNA levels and low or no antibody levels. And if you think that's part of why we're seeing this rise in the rise of maternal or parental to child transmission that we're seeing.
So it's transmission of hepatitis C, and actually a lot of other viruses, but are closely related to viral load. And the higher the viral, I didn't present that study. I had it by too many slides. I had to cut. The viral load greater than 10 to the 6 is closely associated with hepatitis C transmission, as is bleeding, though, anytime during pregnancy. And that's probably, who knows, but it's probably, it could be, the virus can be all over the body, including the genital tract, and who knows, but the mechanism, whether it transverses the placenta or it's ascending, But those are the two major risk factors. I think if women are injecting during pregnancy repeatedly, that's obviously gives you a chance for having, increased chances of transmitting.
Yeah, I think given higher HCV RNA levels in the acute phase and the absence of antibodies could be a risk factor. I'd love to see someone do a study to see if acute infection is resulting in more transmission.
Yeah, that would be, we'd have to find the patients, but there are ongoing studies of treating in pregnancy, or there will be. Capturing acute infection in pregnancy is probably, not oftenly done. I mean, if you did universal testing worldwide, and you use the HIV model, where similarly, if you have high viral loads and you have acute infection, the transmission rate goes way up, and the risk is extremely high. Yeah, that's.
Yeah, I see that someone posted that HCV RNA testing was approved CEPIADS by the FDA, and I do think that represents a really important advance.
Yeah, in the US, but this is a worldwide problem. Most of the infections are not in the US. I mean, yes, we have a large population, but the rest of the world has, 50 million, of which, as I showed you, there's quite a few pregnant women who deliver babies.
There definitely seems like a role for policy in implementing more of that universal testing and preventing transmission and just infection with that treatment capabilities.
Yeah, I mean, that's an area I spent most of my career on, is mother-to-child transmission, and it is a very complex issue because it varies on what trimester and other factors in terms of age and, whether you have C-section or whether you had, how long is your rupture of membrane. There's a lot of factors involved in mother-to-child transmission. But if you use the model that we use for HIV in the rest of the world, meaning all pregnant women are tested and probably should be tested, if they're negative multiple times, then that would allow you to identify the women, and maybe very soon we'll have some studies to demonstrate that it's okay to treat pregnant women early in pregnancy or throughout.
Yeah, pregnant women is the largest population with hepatitis C that are not recommended for treatment at the moment, and we need to correct that, as you point that out. Typically it's about a 3 to 5% risk of transmission from mother to child, with HIV increasing that, as you pointed out. Some of those other cofactors like ruptured membranes and duration of labor and they're sort of weak risk factors. So I think the major point is that we need to screen and treat. There's a few studies going on, as you mentioned already, to see how we can collect the data, mostly on the safety side, to show that these drugs are safe in the mother and the child. And we have a TIP Hep C, a global registry that we're doing with CDC to collect data when a pregnant woman, and the clinician decide that they want to proceed with treatment. They can report the outcomes for the mother and the child. So just it's sort of a little bit of an advertisement for people that are aware of that, to come to our website and learn more about TIP Hep C.
So in the United States, when the epidemic started out, you had a lot more HIV, HCV co-infected pregnant women because IDU was very, that was one of the major causes of transmission. Worldwide, I think it's very similar. Now, recently, with these increases in IDU and, snorting, which has blood, and other ways that you can transmit, women, again, looking at pregnant women, it's recommended that they get tested, but through 9 months, if someone's using drugs, you really need repeat testing.
Yeah, advertising also just the importance of testing in pregnancy. This is in many cases the only contact young healthy individuals have with the healthcare system. And so it is an opportunity to test people who are otherwise not seeking medical care in any setting.
Yeah.
I mean,
Yeah, and the other benefit of treatment during pregnancy is probably Andrea Cox will mention is just how many women fall out of care after delivery, and so they don't get treated for their own health, let alone interrupting transmission to their infant.
right. I mean, what we're trying to do at our site is pull the HCV positive women into our clinic, which is now a co-infected clinic, but also infectious disease clinic. And then the ACTG, it does have some clinical trials, one coming up soon for treating pregnant women. And, you know, I think they need to be in a center where they can have the whole care, meaning not just treat them and let them go, but have ongoing contact, because injection drug use is, you know, a chronic problem. And people go back, they stop and they go back.
Another question for you, Andrea. You notice that there's activation state is decreasing with direct acting antivirals. And yet the percentage of HCV specific T cells in the later stages of infection is very, very small. So I'm curious to know what your hypothesis is about why there's so much less activation when you're probably not removing much Hep C specific T cell activation from that picture.
So you're talking about when they cleared the virus?
At the last study you showed that with DAA treatment, you decreased activated T cells, which is good because you've shown that can enhance progression to HIV, but from in HIV to AIDS. I'm just curious if this is just a global activation state because the percentage of T cells specific for HCV is very small in chronic infection, particularly active T cells, since they're mostly exhausted.
So when you look at the numbers, and we're looking at this data right now, the activation levels overall in HIV are low now. You know, now that they're down to less than 200 overall. But with co-infected, as you saw what I showed, is there are some people that still have high activation levels. And those are the ones that we saw it go down by after treatment. Again, this is not multivariate analysis. It didn't look at viral load. It didn't look at the I mean, there's quite a few. Until we do the multivariate models on a large number, we won't know for sure, but this gives us a hint that, yeah, there is something going on. If you treat someone with with co-infection, treat their HCV, you may make a huge difference in the overall outcome of these women. Now, as I showed you, the fibrosis flattens out. This is a study Phyllis Tian and her group did. And so we think we need to follow for a longer period, and that is because of menopause and fatty liver disease and, NASH and NAFL. So it's very complex because there's a lot of things in play, and you've got to think about all of them, and you've got to tease it out. And then you got to really adjust for all the major factors. But for us, this is a clue. Something's going on. It does look like in, whatever numbers of half of the patients, activation did go down. There's a change in Tregs. And then we have we do have data that terminally differentiated RA positive also goes down. So it impacts maturation as well, that it improves. So it is a very complex study, and it takes it's a lot of work because you got to first do the flow, 13 color flow on hundreds and hundreds of samples, and then you got to sort 9 different samples, and then you got to evaluate how well you've done in terms of extracting RNA and D. I mean, there's a lot involved in it, but we're to the point now where we are running these, and we're going to have some really good data in the next month or two.
Yeah, I
more.
agree. Larger studies would be really helpful.
Yeah.
Jackie, I wanted to ask you, and I also applaud you for incorporating the views of people, both clients and the people providing care to them in your testing. One of the providers mentioned that people might not report positive results, and I can see that that might be a plus in some settings, but for some people, it might eliminate a barrier to testing because there are places in the world where infection is tracked in ways that people who are infected find undesirable, with public releasing of data on infection status, for example. So I can think that might be a plus in settings where people want to test but don't want, for example, an outside group of people outside of their medical providers to know their status. What are your
Yeah,
thoughts?
absolutely. And that data privacy is a big concern. Even in the technology design, we're looking at, oh, well, who does the interpretation? Because we have technology now, smartphones that can interpret that line for you and tell you like that's positive or negative. And then what happens with that? Who owns that information for those that are developing some of the more complex tests that integrate with EHRs and electronic health systems? And so those aspects really inform the privacy and the safety of the individuals that are doing the test. There's convenience involved in having something that automatically sends information, but then there's a lot of things that can go wrong with that. We've opted in this case for a lower tech option so that individuals could do it themselves. But then there's also policy aspects of like, well, is this FDA approvable? I know the first HIV self-test that came out required someone to be on the phone with you, helping you to interpret the test. And because then you can also manage that human connection, but then also there's a linkage to care and notification. So yeah, it's interesting to see both the client perspectives that are valuing that privacy and the providers' perspectives that are really thinking about, how do we get them along the next steps, and balancing those is definitely a challenge for anybody that's thinking about how they might go about implementing and the safety of the individuals that are getting these results.
Yeah, thank you, Jackie. A quick follow-up question. This test for acute infection is very important. We have lousy markers for acute infection at the moment for surveillance or for identifying transmission networks and the like and so I'm just curious, what's your next step in development? Do you have any field trials underway or planned?
That's the goal, is to ultimately get it out and tested in the field. Right now, we're trying to figure out how we can scale up production. The reason we made that little automated device is because it'd been grad students with tweezers assembling things, which is not how they want to spend their PhD, but also there's quality control that we're trying to learn and figure out as we go. So connecting with manufacturers that do this, and also being able to get first usability studies, because we don't want the test to fail because people didn't know how to use it right, and then out into really doing those field trials. So yeah, it's definitely in the plan. We want to get things out of the lab. They don't do much good sitting with me.
Well, it's a very important area. Thank you very much. And thank you also, Andrea Kovacs, for all the basic science research. I'll turn it over to Raul and Wilson to say farewell. Again, all of this information will be available on our website, globalhealth.org, and I think there may be an evaluation popping up here for you to fill out. Wilson, Raul?
Well, thank you very much. Clearly, you two are at the forefront and at the cutting edge of research, both in terms of device development and sort of product development that can really have an impact on population health. And certainly even in the questions that we got from the field asking us about, well, what about people that don't qualify for the new point of care system out of Cepheid? Might there be room for other devices that can really help fill in some of those gaps? Of course there is. The other area is we're really keen to understand the impact on women's health. That's a major NIH priority right now. So Dr. Kovacs, congratulations on some very important work that will really shed the light on both care for HIV and HCV, but I think more generally around an aging population that we need to have much more research on. And with that,
thank you, Wilson. Wilson,
very
I
much.
think that you said everything, and I thank all the participants. I thank the organizers, Dr. Ward, and the panelists. It was a fantastic webinar, and hope we get more questions, and you will be able to look at all this webinar online. Thank you.